Good afternoon. Let's get started until while people are still joining. So it's my pleasure to introduce today's speaker, Ben Ho Park, who is professor of medicine at Vanderbilt University Medical Center. He's also a deputy director there and director of precision oncology and division director of hematology oncology. And Ben completed his MD PhD at University of Pennsylvania, and then continued his residency training there as well, and then did a postdoctoral fellowship with Bert Vogelstein's lab at Johns Hopkins, and we just barely missed each other. He started when I left the lab, and he stayed at Hopkins until 2018, going up the academic ladder and leading a very successful research lab in focusing on translational breast cancer research. And Ben has been a pioneer of using somatic cell engineering for studying the function of tumor suppressor and oncogenes in breast cancer in particular. And he started doing this before CRISPR was very much available. So, at that time, it was much, much harder to generate those knockout, knock-in cell lines. And he continued, his work. And then he moved to Vanderbilt and took on these leadership roles a few years ago. And today he will present Subclonal Evolution, a touching story, a very intriguing title. So we're looking forward to your talk, Ben, and thanks for joining us today. Thank you.
Thanks so much, and thank you for inviting me and hosting me, Nelly. Yeah, for those who know me, you know I love a good pun, and so that will become obvious as this talk goes further. I also oops, hang on a second, I'm going to share screen. And I also was just saying to Nelly that I don't think I'm going to take up the whole hour. The rule of thumb is one slide per minute, and so I have about 33 some, and consider it an early Christmas present or holiday present, because I'd like to say nobody ever complained of a talk going too short. So yes, this is a new story kind of in our lab, or a new area of research that we've been working on. As you'll see, it was really inspired by some of the work that Nelly did in her lab that was published a few years ago. Before I begin, though, I do want to kind of pay homage and remember Dr. David Livingston. I know it's a huge loss for your institution, but I'm sure many of you recognize it's a huge loss for the whole kind of cancer industry, if you will. David was one of actually, I think he was the chair when I was at Hopkins of our external advisory board for our CCSG, our core cancer center support grant. And that's where I really got to know him. And he was just a wonderful guy, role model, super smart, would always have very thoughtful, insightful suggestions. And so I really was shocked to hear the news, as you all were, and I really miss him. He was really someone that I looked up to and actually helped me in a lot of different ways. All right, I think this audience probably recognizes that tumor heterogeneity is a very well accepted aspect of cancer. And when we talk about tumor heterogeneity, it's more than just that within patients that we see cancers are different from one another, but even within a patient. And even that takes on several layers of complexity. We often talk about intra-tumor variation, shown here on the right-hand side of the slide, where we have different subclones that actually make up the whole mass, which we call the tumor. But as cancers metastasize, those subclones can shift in nature and new subclones can appear and evolve. And that's something that we've known for pretty much decades, that in fact cancer is a slippery disease, meaning that the DNA is unstable. And so when one cancer cell divides, it doesn't beget identical twins, so to speak, but in fact they're a little bit different. And this is what makes cancer so difficult to treat and cure. When we think about how we're using systemic therapies, it really is dependent upon making sure that we can eradicate every single cancer cell. And with tumor heterogeneity, that's very, very difficult. So oftentimes this will lead to recurrence and metastasis. Now, as I mentioned before, prior work from Nelly's lab really led us down this path of starting to investigate tumor heterogeneity in breast cancer. And so Nelly's group published this really great paper. When I first read this, I love acronyms, and so STARFISH, or specific to allele PCR fish, was something that Nelly developed in her lab. And so this paper, in situ single-cell analysis, identifies heterogeneity for PIK3CA mutation and HER2 amplification in HER2-positive breast cancers, really caught our attention. And you can see here in the diagrams, I'm not going to explain this because it's always daunting to explain other people's work, especially when that PI is actually the host of this. But the end result of this is that this technology or this technique allowed Nellie's group to look at in situ hybridization techniques, but using PCR that was mutation specific. So we hadn't really been able to do that in the industry before, allowing you to look at a tumor tissue and say, oh, this is a mutant allele, this is a wild type allele. And the results were really striking and provocative. They suggested that, in fact, PIK3CA mutations were sometimes in cells that did not have the HER2 amplification and vice versa, that those are independent events and that can actually lead to a fitness advantage. And sometimes they are in the same cell. So it really depends on that patient's individual tumor, whether they're separate or where they're combined. One of the other reasons that we got interested in is we published this work a number of years ago now, and I'm embarrassed to admit I forgot where. I think it's in JCI Insight. But one of the things that we did was a rapid autopsy series. It was very small. I think it was about 5 patients, and we got 3 METs and tried to get the primaries where we could. And so we had 3 METs minimal for each patient, and we did next-gen sequencing. And we looked at, across the METs, if they had a mutation or a variant, as described here, VAR1 or VAR2, how they compared between METs within the same patient. And so the diagram that we came up with, this was work done by Sarah Whalen, who did this collaboratively with our lab. 0 means 0% allele frequency, 100 is 100%. And so let's say this is MET1, MET2, MET3, all from the same patient, and this particular variant was about 50% in all, shown here 50, 60, 40. There were some cases, though, where there were unique or private variants that were present in just one MET and the other. And again, that's probably not so surprising, and that's depicted here. And so you're probably familiar with these types of Circos plots that, look at CNV, copy number variation, loss of heterozygosity, allele frequencies, et cetera. In some patients, what we actually showed using the diagrams that I showed before, that in fact there were a lot of similarities between METs. Again, maybe that's not so surprising, but it's hard to dig into this analysis. There were some where the allele frequencies and what we probably would call now subclonal architecture seem to be very, very similar, if not identical, between the 3 METs in an individual patient. Now, we had done what we call subclone analyses with certain software programs that many of you are probably familiar with, PyClone, SciClone, et cetera. And at the time, to me, it seemed like this was not real. And so we chose not to publish it because we weren't 100%. These were relatively new kind of bioinformatics tools. But I always stuck that in the back of my brain. And again, when Nellie's paper came out, I thought about, wow, this is perhaps telling us something, that subclone architecture is not in fact random, but maybe because of whatever factors, the fitness that allows a tumor to grow as a MET and or maybe drug resistance and evolution has the effect where subclone populations will self-assemble, if you will, and kind of grow in a way that optimizes growth of the mass, but not necessarily one subclone versus the other per se. Just a little bit of background about HER2 and breast cancer. So HER2, I think many people in this audience are already aware of this, is overexpressed or amplified in about 15 to 20% of breast cancers. This leads to proliferation, survival, and motility, or increased motility. The clinical designation of what we call HER2 for breast cancer, at least, is based upon amplification or protein expression or overexpression. And so we use either immunohistochemistry or FISH to evaluate this. The reason why this is important is because HER2 breast cancer, positive breast cancer, used to be one of the worst, if not the worst, prognostic categories of breast cancer, or subtypes, I should say. But now it's actually the best because of targeted therapy. So this is really one of the first success stories in cancer in terms of targeted therapies and discovery of a target, and then the development of a drug that really changes the outcomes. Now, PIK3CA mutations, as I was mentioning earlier from Nellie's work and others, has really shown to be probably, maybe up to 50% of HER2 positive breast cancer. And there's some biological rationale and even some laboratory work and maybe some clinical work to suggest that if you have PIK3CA mutations, you may not be as sensitive to HER2 targeted therapies. This kind of made sense and was around the same time that we discovered as a field that KRAS mutations predict for resistance against EGFR-based antibody therapies. And again, looking at the pathway, that makes sense. But this has not been such a black and white issue in terms of definitively showing that PIK3CA mutations lead to drug resistance. Now, we started focusing more on HER2 missense mutations because of some of the work that Nellie mentioned. We were very interested in doing kind of knock-in isogenic types of somatic cell modeling. And so when we looked across various databases, HER2 mutations are the minority in terms of breast cancer. So most are, again, amplified, as shown here on the red bar in this oncoplot. And then there's a small minority that actually have mutations or missense mutations. This is in contrast to the PIK3CA. And again, there is an even smaller fraction where you have overlap, where you have both mutations, PIK3CA and HER2 in the same cancer. The thing about HER2 mutations, though, is there are hotspots similar to PIK3CA mutations. And when we see hotspot mutations in cancers, at least I think about the fact that these are probably not by chance, that they're being selected for because they have some sort of functional consequence. And sequencing studies have really verified that there are kind of clusters of hotspots in the kinase domain region as well as the extracellular domain. And certain cancer types seem to have more of one or the other. In breast cancer, you seem to have more kinase domain mutations. As an opposite example, in brain cancers, you see a lot more of the extracellular domain mutations. People have done a lot of studies overexpressing these mutations and showing that they have transformative phenotypes. But it's actually not that common to have a missense mutation that is in fact also amplified. You tend to have more of an amplified HER2 that's wild type or just a single allele that is a missense mutation. And in some ways that makes sense because you don't necessarily need that biologic redundancy. There are probably some examples where people have shown that you can get amplification of a missense mutation that might lead to drug resistance, and whether that is acquired or de novo, I think depends on the clinical scenario or situation that people are looking at and getting these samples. All right, so we got also excited about studying these because we knew that there were clinical trials going on targeting these. First looking at tyrosine kinase inhibitors, and subsequently later there's now been a lot of studies, mostly smaller or anecdotal case reports that seem to suggest that even antibody-based therapies, particularly with antibody drug conjugates like in HER2 and Kadcyla now, can actually afford benefit for HER2 missense mutations that are found in various cancers. But this was a study put out by the Memorial Group a number of years ago in Nature called the Summit Trial, and it really did show that you could use a drug called neratinib, which is a tyrosine kinase inhibitor, in patients with HER2 missense mutations. They also included HER3, but there weren't that many patients who had HER3 missense mutation, and you didn't really see a signal using neratinib, which is a pan HER family member inhibitor. I'm not going to get into the weeds of the details, but suffice it to say, this wasn't necessarily across all tumor types where they saw actual benefit. It was mostly in breast cancers. And so for our purposes and what we wanted to study, that was still pretty exciting to see that, in fact, these HER2 missense mutations could be targeted with kinase inhibitors. Whoops, sorry about that. So a number of years ago, as Nellie had mentioned, we were doing gene knockouts, knock-ins, somatic cell gene targeting is what we used to call it before CRISPR-Cas made it kind of cool and easier to do. We kind of did it really the old-fashioned way, and some might say the crazy way, but this is a technology that I learned in Bert Vogelstein's lab when I was a postdoc and brought it with me to my own lab at Hopkins in the breast cancer program. So we generate a whole library of different cell lines. Our workhorse is the MCF10A, which many of you are familiar with. This is a non-cancerous human breast epithelial cell line that's been spontaneously immortalized. And the variants that we've had are remarkably genetically stable. The diploid is almost normal. They do have a homozygous deletion of 9p, which probably accounts for the spontaneous immortalization. And they really seem to hold their own in terms of their karyotype. We've done a number of studies where we did single allele knock-ins of mutant PIK3CA, as well as HER2 missense mutations. And this was probably one of the most interesting ones because we have found that when we did PIK3CA, it imparted some cancerous phenotypes to these cell lines, most notably that they can now grow without the addition or putting in exogenous growth factors like epidermal growth factor or EGF. On the other hand, most of our knock-ins and knockouts that we created have very minimal phenotypes in the cell line and a companion cell line that we call the H-terdimex that we got generously from Miles Brown lab. And so it's always been interesting to us because what Dan Zabransky showed, who was an MD PhD student in my lab, is when he knocked these mutations into 10As and even MCF7, and I'll go into this a little bit about the cell lines, that in fact the phenotypes were minimal to none. It was really more about just showing some subtle increase in biochemical signaling. But when he combined them into the same cell line, that's where you could really see some new, maybe even synergistic effects. An example of that is here, probably the biggest phenotype that we saw when we did the double knock-ins, which is the DKIs. So here you're looking at the parental MCF10A cell lines. This is the parental control, the single PIK3CA knock-in of the exon 9 hotspot, a kinase domain knock-in of HER2, and then the double knock-ins. And these are, this is work done in collaboration with colleagues at Johns Hopkins, putting it through these capillaries at different diameters and having some sort of gradient attractant, and we could actually see increased migratory phenotypes, and hopefully this movie will work. And as you can see here, this is the parental, this is the single PIK3CA knock-in, the single HER2 knock-in, and then you can see more cells going over to the other side with the double knock-ins. And that was true both the 50 micron and the 20 micron chambers. Again, hopefully you can see that going. And these were quantified. We also did this in a second cell line, MCF7. And so just to remind everyone, MCF7 natively has a PIK3CA exon 9 E545K hotspot mutation. Turns out there's actually 3 alleles of PIK3CA in MCF7, 2 of which are mutant, 1 is wild type. Josh Loring, who was a postdoc fellow in my lab, actually corrected that, and then we had an MCF7 that was wild type for PIK3CA, and then Dan took that cell line and then put it into, put a HER2 missense mutation into it, as well as into the parental. So just for sake of clarity, what you're really seeing here is just exon 9 and a double knock-in, so to speak, even though it just says HER2, because this is actually derived from the parental. But you can see here, once again, that these cells that have both HER2 and PIK3CA really have a phenotype where they migrate faster and maybe better, one might say. I've always found this to be really weird too. I think there's a name for this, like an oncoprotein, but it looks like this cell is passing off a little bit of food or something, or a basketball. I don't know if you can see that here, and I've still never figured out exactly if that has any physiological relevance or consequence. So if anyone in this audience has some thoughts about that, I'd be happy to hear that. All right. So again, just for sake of clarity, because these are some of the terms or nomenclatures that we're going to use for the rest of the talk, we have 2 real panels of cell lines, MCF10A, which is again the non-tumorigenic human breast epithelial cell line that's spontaneously immortalized. It is EGF dependent, meaning you normally have to add or supplement EGF into the growth media to get it to grow, and that's the wild type. We also use as a control a SNP. We were working on this introducing mutations in a gene called SF3B1, and one of the things that we did is introduce a silent SNP into this, and that's kind of because it goes through the same gene targeting process, and it actually goes through the single cell clone process. So that's really kind of an ideal control. We've also made Josh Loring, when he was in the lab, an AKT1 knock-in. Again, doesn't really have much phenotype, still remains EGF independent, and then I told you about the HER2, which is also EGF independent. So really it's this guy, the PIK3CA knock-in, that has a phenotype of EGF independence. On the other hand, these are cancer cell lines down here, MCF7. I already told you about that PIK3CA is naturally mutated, and there's 2 copies of that and 1 copy of wild type in the parental. The wild type is the corrected version, where now it has 3 wild type alleles, and then the HER2 takes the wild type corrected and introduces the HER2. And we did also create a double knock-in of the HER2, but that's not for MCF7, but that's not going to be shown here. So what we decided to do after Nellie's work came out was we had a thought, well, you know what? We have a very simplistic but elegant model to study this, where we could just put mixed cells together and see what happens. And so that's what we started to do, not really thinking much would come out of this, but we put them in co-culture. And we're talking here about either MCF10A or MCF7, but you just mix them at 50-50. We tag them with RFP, GFP for the rest of this talk. The RFP is going to be the HER2, and the PIK3CA is the green cells. And then we harvest the cells at various time points. We look under the fluorescent microscope. We grind it up and make genomic DNA, and then we do droplet digital PCR to do absolute quantification of the alleles of mutant PIK3CA versus HER2. The artificial mix is this kind of moniker that we came up with, and we just grow the culture separately in various conditions. And then we do the same thing. We look under the fluorescent microscope separately in these, but then we actually will mix them to grind up the DNA so that they go through the exact same process as the co-culture, and then do the same type of droplet digital PCR analysis. What we found was very interesting and somewhat surprising, and that's shown here in graphic format, but then here in pictures. If you look at the top, this is the MCF10A system, where we've mixed the single knock-ins of HER2 and PIK3CA. You can see in the artificial mix, it's kind of what you would think. So what we're looking at here is the fraction of HER2. Now, what you have to remember is this is in a media condition without EGF. So the HER2 cells will just basically arrest and not grow, whereas the PIK3CA cells will take off. So I'm not showing the PIK3CA here, the fraction, but you can imagine in this particular artificial mix, the PIK3CA is going to go up. On the other hand, what happens with co-culture, the HER2 cells do something interesting. They start growing. Even though there's no EGF in the media, they start picking up and taking off. That's in stark contrast if you use a wild type mixed with a PIK3CA. And so again, you get exactly what you would have thought, where the wild type cells are just not going to grow, even in the presence of PIK3CA cells grown in co-culture. And this is also recapitulated in the MCF7. Just want to point out here that, remember, MCF7 as a cancer cell line doesn't require growth factors, but you do see in the system with the co-culture a growth advantage to the HER2 population when you actually do it as a co-culture versus just an artificial mix. And again, if you use wild type HER2 cells, then you don't really see any appreciable difference between co-culture and artificial mix. It's much easier to visualize this down here, and we put early passage and late passage. So early passage is on the top, late passage is on the bottom, and I think you can see a pretty striking difference of what happens when you use HER2 with PIK3CA. The red cells, again, which are HER2, become the predominant population with late passage. That does not happen with the wild type system. And here in the MCF7, you see a very similar phenotype with long-term passage. You actually see the predominance of the HER2 population in red and very few green cells. And that's the opposite if you just use the wild type. It just seems to be about 50-50. And so our next question was, well, is this PIK3CA, is it mutant HER2, or is it both? It turns out it's probably both. So we first did PIK3CA with AKT1. And you can see here, it looks like just the wild type. So the AKT1 cells will not grow when grown in co-culture with PIK3CA. We've also had some cell lines, one of which hasn't been published yet, but we're getting ready to submit it, where we've knocked in Notch1 mutations. And in this particular mutation, we can actually get growth factor independence, or EGF growth factor independence. So if we put that with the HER2, one might think that's what's going on and doing it, but in fact it's not. So it's more than just EGF independence per se, because you can see here from the red RFP representing the HER2, that in fact you don't really get the predominance of HER2 as you do with when you co-culture HER2 with PIK3CA. What was more striking and surprising was we asked the question, well, what happens if you start with different ratios? Before all those other experiments I told you about, we're starting with a 50-50 ratio. Here we decided to do the experiment of starting off with 90% PIK3CA and 10% HER2, and then conversely 90% HER2 and 10% PIK3CA. But it doesn't seem to matter. If you take these cultures out long enough where you just keep serially passing it, eventually the HER2 population becomes the one that takes over. And again, just to remind, this is in the absence of EGF. We do that with MCF7, and we see, again, pretty similar results recapitulating what we see in the 10A cell line, and that is with prolonged culture, the HER2 population is the one that becomes the predominant one. And then we asked the question whether or not we could recapitulate this in vivo, and the answer is yes. And so only the MCF7, I think, as many of you know, are tumorigenic. The 10As are not. Even when we put in PIK3CA, they end up not becoming tumorigenic. But here's just a growth curve to, the tumor volume curves to show the actual data. But suffice it to say, it was pretty much does recapitulate what we saw in culture. And we took out the tumors, and so they're still tagged GFP and RFP. And again, you can see that you can have with the HER2 mixed population, predominantly the HER2. I apologize, these were somehow the labels didn't come over. This is going to be the wild type control. All right, and people also ask the question, what happens if you grow these in 3D in Matrigel? And pretty much the same thing. This is a little bit harder to see, but hopefully you can. Here we're looking at a co-culture and HER2 with PIK3CA. The nice thing about this in some ways is that you actually separate out each individual cell that grows into like its own acini. But I think you can appreciate in the overlay where you really see red growth starting to take over is only when they are mixed and touching green cells. And here, a little bit going on here. but hopefully you can appreciate that. The um, other thing that we were curious about and asked is, well, how is this happening? Is this potentially just cytokines? What's being, eluded, if you will, or given off by the mutant PIK3CA10A cells? Turns out it really doesn't seem to be a soluble factor. And when we did transwell assays, which many of you are familiar with, where you simply put cells on the top versus cells on the bottom so they can't touch, but they share all the same media, there really was no appreciable difference in wild type versus HER2 growth when you layer them with PIK3CA, and it didn't seem to matter if it was top or the bottom. And so it didn't seem to be it was a soluble factor. What we ended up doing to really show that this is a, quote, touching story is doing time-lapse video photography. And I put this on here because sometimes the video doesn't work, but I'm just going to go to the video because we went ahead of time. And this is, again, in the 10A system. What I will point your attention to is if you look at these cells in the red, they may divide once or twice. These are the HER2 cells, and in the absence, so these are just plated, and that's why they still may undergo 1 or 2 rounds of mitoses, but then eventually they'll arrest. On the other hand, every red cell here that is touching a green cell, you'll see with time it ends up starting to grow. And so hopefully this will work, yeah. So again, looking at the red cells touching the green cells is ultimately what happens. And these guys just kind of hang out, and then at the end, this one will start touching, this one will start touching, and they'll start taking off. So this was our first real clue that, in fact, this wasn't something necessarily, at least, just with soluble factors, but at least minimally had to have some sort of cell-cell contact or interaction. Sorry, I love looking at this video. So what was going on here? And we decided to kind of do the logical thing, and that is to look at gene expression changes. And so we did flow, and we looked at co-cultures as a total, and then flowed red and green, and then looked at RNA expressions for individuals. And we pulled out, as one usually would imagine, a whole host of different genes that were up and down regulated. One of the things that was striking, though, was this gene, FN1, which encodes for fibronectin. We had worked with Carlos Arteaga's group a number of years ago and published a paper that 10A cells and PIK3CA may make more fibronectin, or at least it seemed to be make more fibronectin. So we thought that was a very interesting finding, and one, it felt good that we could reproduce our own data, but this was something also that we saw in the co-culture, and it looks like from, if you look at this plot, that in fact if you measured fibronectin in the co-culture, so this is both PIK3CA and HER2, and you have to remember that these guys now are predominantly HER2, that you see a lot of fibronectin expression. And that was really interesting to us. The MCF10A, using the MCF10A system, one of the things we decided to do is, well, can we recapitulate this phenotype if we actually add to the HER2 cells fibronectin? And so this is an experiment where we just use the wild type cells alone, the HER2 cells alone, and the PIK3CA cells alone, and either use vehicle control or added fibronectin exogenously back to the culture. And you can see here that, in fact, in the HER2, you can see this appreciable difference in cell type, or in cell proliferation, I should say. There is a known receptor for fibronectin. Many of you probably know this. It's one of the integrins, and there is a blocking peptide, RGDS, that one can either synthesize or buy commercially. But people have shown that you can use this RGDS peptide to block the interactions of fibronectin and its receptor. And so that's what we tried here, and that, again, we seem to show that, in fact, that particular peptide can block the effects of co-culture of HER2 and PIK3CA. And that's shown here graphically, or I should say photographically. This is the vehicle control, where you can see a lot of red cells, and this is the peptide, where it doesn't block it completely, nor would I expect it to, but it is dramatically different compared to the vehicle control. So of course, coming from the Vogelstein lab, if you really want to prove this, you got to knock it out. And this was right around the time CRISPR-Cas really started becoming hot, so it was made for a much easier proposition. And so we did that. Swati was a graduate student in my lab, Karthikeyan, who came in and then did the knockout in both 10As and MCF7. And you can see the knockout sequences here. These are the antibody conformations using Western blots of the 2 independent knockouts that we had. And this was a photograph that Swati took when she did an initial co-culture of PIK3CA, which has the fibronectin knockout, as well as a HER2 cell. This is the one that's still labeled red, so you can see it right here. Now, the gene knockout, I'm getting to that, will abrogate the phenotype, but we also showed in culture that there's really no appreciable differences in cell proliferation between the fibronectin knockouts and the parental cell lines, and that was true for both MCF10A as well as the cancer cell line MCF7. And again, you can see here now in the co-cultures that this is something where we don't see the phenotype anymore. I guess that's, wait, hang on, knockout. I apologize. We don't have the red green tag here, so maybe it may not be as easy to see. Oh, wait, I'm getting this screwed up. I apologize. This is just looking at the cell's morphology grown a single culture of the PIK3CA mutant versus the PIK3CA mutant with the FN1 knockout. But this is the one that really shows the difference of fibronectin being knocked out in PIK3CA. And so now you see a situation just like you would with wild type. The co-culture of PIK3CA fibronectin 1 knockout with HER2 mutation does not confer growth advantage to HER2 anymore. And so that's seen here. We do a 50-50 mix at early passage. By late passage, normally you would see the red take over, but because PIK3CA now has fibronectin knocked out, you don't see that phenotype. So the PIK3CA cells are still growing. We did not label these green because these are, again, the PIK3CA fibronectin 1 knockouts. But we do still see in the HER2 minimal amounts of red, and that was recapitulated again in the MCF7 cell line panel. And then what about actual human tumors? As I said before, missense mutations of HER2 are not that common, but we actually just looked across cBioPortal and various databases. And as you may expect in the gray areas, most of the time you see a pretty good concordance of the allele frequency of PIK3CA and HER2. But there are these cases way off here to the left where you see high numbers of HER2 missense mutants, allele frequencies, and lower numbers of PIK3CA. So does this happen? I think that it can happen in terms of the skewing of the alleles. And remember what I said before, it's rare that you do actually get amplification. So as near as we can tell, we believe that this is more of a kind of subclonal architecture, if you will, but we can't prove that with the existing data that we had at our disposal. On the other hand, we also wanted to ask the question whether or not fibronectin 1 was ever really expressed in human breast cancers that have PI3 kinase alterations, mutations, or otherwise. So we had some samples that we had from prior studies here at Vanderbilt, and we looked at this and really want to thank Melinda Sanders and her team and our translational pathology core to help us with this. But the fibronectin 1 highs, there was a couple in the ones that actually had mutant PIK3CA. When we stained at those, Melinda has a very high standard of what she's calling high, and I think you can really appreciate the difference between what we were calling fibronectin 1 high or fibronectin expression versus being negative. So it was really all over the place. It was striking. We wanted to get higher numbers though, and so we kind of mined the database, and this is the Metabrick database, which many are familiar with, and we looked at just tumors that had increased expression of fibronectin, again, the gene being FN1, and we looked at all PI3 kinase alterations. And so we actually found a fair amount, about 82, if you include both mutations mutations in PIK3CA, maybe as well as amplification, but also other PI3 kinase alterations like PTEN loss and PI3 kinase beta. So we don't know how common this is. My gut tells me it's not that common, and in some ways I'm grateful for that because cancer is complex as it is. But this made me really kind of think about, wow, cancers are really, really complex sometimes, because who would have really thought that you would have these independent subclones that can communicate with one another, and what does that really mean for that patient's tumor and targeted therapies? So in conclusion, I hope I've at least, maybe if not convinced you, made you think about in a provocative way that perhaps tumor heterogeneity is not random, but in fact is selected for. That subclones don't just have a certain population in a tumor mass because that just happens to be the one that's the lowest amount represented. That in fact, there's probably some local eco-environment, if you will, that selects for an architecture or subclonal architecture that is not random, and that could be from what is in that patient's body at the times in terms of drugs, or just how they set themselves up in the liver versus the bone or the lung. This probably depends upon the genetic makeup of the subclones and the selective pressures to become metastatic, and again, the resistance or response to therapies. That fibronectin really mediates, at least in our hands and models, the specific interactions between mutant PIK3CA and mutant HER2 cells, and that in the future, one of the things I'm doing here is directing precision oncology, which, I was also heavily involved with at Hopkins. But I started these molecular tumor boards, both at Hopkins and which continues to go forward, but also here at Vanderbilt. And when we came across a subclone of PIK3CA or a 5 or 10% allele frequency, or any oncogene for that matter, we would deprioritize that because our thinking would be kind of traditionally you'd say, well, why would you go after that if you have something else that's at 90%? And so I don't know if that's the right way to think about it anymore. And so that's what makes it really challenging. Again, thankfully, this seems to be the minority of cases and not the majority. So it's not something that I lose a lot of sleep over, but there are instances where we really scratch our heads now and think about, is that in fact the right answer? And that leads to this bullet point that maybe targeting subclonal populations would be the better thing to do, or at least doing it concurrently or sequentially. And then that also brings the point that fibronectin, in fact, by itself may be a target for future therapies or maybe even some sort of prognostic marker. This does not happen in a vacuum, as any of you who run labs know, and so there's a ton of people to thank, both past and present. The two co-authors who really led this study are Ian Waters, I misspelled his name, Waters with an S, and Ian was a PhD student in my lab at Johns Hopkins, but then decided to go to medical school and is now a medical student in Chicago. And Swati Karthikeyan is a PhD student in my lab who graduated and is now a postdoc at Stanford University. Dan Zabransky, I mentioned, did the whole HER2 knock-ins and double knock-ins when he was a MD PhD student in my lab, and Braca did the rapid autopsy genomic genetic analysis when she was a PhD student in my lab. And again, a whole host of other people who helped out that I don't really have a lot of time to go through, as well as the people who have funded all of this work. And I think with that, I can stop share. And
okay, thanks, Ben. It's really nice to see that we stimulating people to do experiments, and that's one of the most exciting things about science. And I think you're totally right about there is a subset of tumors where we see this low allele frequency, and it must be doing something. So.
Yeah.
This is one area that we disagree with Bert, because Bert told me that there cannot be a selection for a combination of clones. He still likes to believe that very
I know.
Clean evolution.
He doesn't believe any of this.
Yeah, it's okay to disagree with him. So
yeah,
it's fine. All right. So I have a couple of questions, but let's wait for the audience. And
okay.
There's a question by Dr. Taivo asking do you think the microbiome could have a role in the heterogeneity and the tumor microbiome in particular?
Yeah, no, absolutely. I think, the microbiome is still a super hot topic, and I think that unquestionably it can have roles in tumor evolution, response to therapies, immunotherapies. I'm sure many people are now aware of are also influenced by the microbiome. So I absolutely think that could be the case. What I don't know, and maybe this is what you were alluding to, is how does that affect, or does it affect, this type of phenomenon where you might have two independent subclones, and does that actually somehow lead to an interaction? One can imagine with the microbiome that you have inflammatory processes that lead to maybe upregulation of receptors or stickiness, etc. So, that's something that we've thought about. It's hard to know how to address that in a definitive way. And so we haven't really invested a lot of our research yet. But certainly if there's any ideas, then we'd be happy to collaborate.
Yeah, I mean, there are some quite striking papers that's hard to believe that you actually have bacteria, and particular bacteria, and breast was one of the one that seemed to have a very active and mixed bacterial composition. So I have a couple of other questions myself. So one of them is, fibronectin is secreted. So could you provide fibronectin into the media or in the cells, plate them, and then kind of reproduce the effect? Meaning, is this only fibronectin, or fibronectin is actually a marker of a shift to a more mesenchymal phenotype?
So I apologize, Nellie. I actually did show that, but I think probably I didn't show it well. Let me just share the screen. Oh, I can't.
You need the co-card. I mean, you need the
so we put exogenous fibronectin in, and you can actually recapitulate, and it happens only in the HER2. It does not happen in the 10A as you would expect. So
Okay.
you have to just put it on the wild type 10A, I should say. You have to, it works on the HER2, and it doesn't work on the PIK3CA in terms of increasing. So we did PIK3CA by itself, with and without fibronectin,
Mhm.
HER2 with and without fibronectin, and then 10As, parentals, with and without, or controls, I should say. And
Mhm.
only the HER2 actually responds and grows.
Okay, that's very interesting. The other thing what I noticed, I mean, based on this time lapse, I don't know how accurate my eyes are, but it looked like that the green cells around red started growing, but then
Yeah.
it looked like that the green cells touching the other green cells that touching the red cells also started growing. So,
Yeah, although it's interesting, I think you're right, but this is a fairly early passage. And what happens at the end, I don't have a picture of it right
mhm,
now, but um, when it's 90% HER2 and 10% PIK3CA, if you really look down with like a
mhm,
confocal or anything, you can actually see that the PIK3CA cells are, and maybe this is part of the fibronectin that they're secreting, but it almost looks like they're neurons now, where they've actually got these projections, and it's
I
like
see.
they're like a net
Uh-huh.
covering the HER2 cells. It's the most fascinating thing in the world to see because if you're unless you're really looking for it, you're not going to see it. So it is more than just touching. They actually kind of change their morphology with time, and it looks like if they're not, got their tentacles, for lack of a better term, all around the HER2, the HER2s are not going to grow because at some point the HER2s, you can imagine,
Mhm.
if there's not enough PIK3CA cells, they're just not they're going to stay there and become static.
Yeah. Okay. And then one last question is like you remember like Carlos Ortega generated these double mutant mice, the HER2 transgenic, PI3 kinase transgenic, or double transgenic. And
Yeah,
then he also noted that in the double transgenic, they did observe cells, even though it was a transgenic mice, but they did have cells that didn't have both mutations.
yeah,
Then they looked at the transgene, and then those um, tumors seem to be more metastatic when you had both oncogenes in the same tumor. So, I mean, coming back to the fibronectin, fibronectin is very common target of TGF beta.
yeah.
Do you see any kind of sign of EMT or PI3 kinase, certain PI3 kinase mutations shifting the phenotype?
Yeah, we did, and I had that in the gene expression array that we saw. So
Mhm.
there are some clues that it may be EMT and shifting. What we couldn't figure out though was like how many genes makes it enough to say that it's EMT, and then
Yeah.
also in certain conditions you might actually see that in just the PIK3CA anyway. So
Mhm.
I think it could be a possibility, but I can't tell you that definitively.
Okay. All right. Sounds good. Very nice. So looks like there are no more questions from the audience. Then thank you so much
All right.
for this very nice talk. So now we know what the touching is. So okay. All
All right.
right.
Thank you so much,
Thanks,
Nelly.
Ben.
Thanks,
Thanks. Bye
Claudia.
bye. And thank you everyone for joining us today.
Take,
Thanks. Bye
take
bye.
care. Bye bye.
Bye bye.